Welcome to the today's episode on the Academy of Bronchoscopy and Yashoda Hospitals webinar series, monthly webinar series. Today's topic is on technology and new product introduction. So we all know we all do EBUS in the intervention pulmonary departments, and then we are very familiar with the rows what we do for rapid on-site evaluation of the samples what we get from EBUS TB and needles. Today we will be introducing you a new technology that is in the process of development, that is automated sample preparation platform for FNA samples from ASP Health. And today we have a very special guest, Dr. Subramanian from the United States, and he's an accomplished scientist, entrepreneur with more than 10 plus years of experience in translating academic research into commercially successful technologies. He has co-founded 3 healthcare companies that are focused on the areas of cancer diagnostics, cytopathology, and robotic surgeries. He's currently a chief commercial officer at ASP Health and a research faculty at Northwestern University. Dr. Subramanian has more than 60 publications in internationally renowned journals and has more than 25 granted pending US patents. And he received his PhD in bioengineering at Northwestern University and MBA in marketing and finance from Kellogg School of Management. He has won numerous business and career awards. His recent award was the Top Healthcare Visionary Award at the Health Conference held in Dubai on the June 20th of 2023. So with this introduction, I would welcome Dr. Subramanian to share his thoughts and how he's going to help us in improving our speed of diagnostics in the operating rooms, especially in the endoscopy suites, both gastro and pulmonary sections. So over to you, Dr. Subramanian. And thank you very much for accepting our invitation to present in our webinar.
Thank you so much, Dr. Harikishan, for that introduction. Thanks for that. And let me go ahead and then share the presentation over here. Okay. So as Dr. Harikishan mentioned, I'm the Chief Commercial Officer of ASP Health and also research faculty at Northwestern University. So ASP Health is an early stage company that is based out of Chicago, Illinois, in the United States. And we have been specifically focusing on streamlining the entire process of rapid on-site evaluation. So our first product here, which I'm going to be talking today, is on the automated sample preparation device. So before I go on to talk about the device that we have developed, etc., so the question is, what is happening with the rapid on-site evaluation today, right? So we have numerous publications and there is a lot of information that shows that rapid on-site evaluation is critical during the TBNA EBUS procedures, right? There is a lot of data that shows that it increases the diagnostic yield and it decreases the number of needle passes that is being done. And most importantly, it reduces the complication rate and overall cost of the procedure. Even though it has a lot of these advantages, what we have seen, at least based on the data that is available in the United States, is there is close to 30 to 40% of the clinical sites still do not perform the rapid on-site evaluation. And even in those sites that perform rapid on-site evaluation, it's not being done on all of the procedures. It's being done on some of the lung procedures, and it might not be done in thyroid, or it's not done in gastro, etc. So why is that the case, right? So, and this is where the challenges on the current implementation of the ROAST comes in, right? It is a highly variable process. And so it leads to very high variability in sample quality, right? Because, for example, as you might have seen in your own institution, you might have one person coming in who's really experienced in making rapid on-site, and that person goes on a vacation, and then you do not have anyone else who can actually take it over, or someone else with less training comes in, which leads to the variability. And it is a pretty cumbersome process, right? Because you have the sample coming in, and then there is a heavy flow of sample that's happening, and then someone is manually preparing it. And as the sample is getting prepared, someone else, the next set of sample comes in. And this is something that we have seen in many clinical sites here in the United States, where as pulmonologists, you are extremely fast in going in and then taking the FNA, but then we see that the cytotechs are in the middle of their staining process, and then the new sample comes in, right? So, and then the most important thing is it's a pretty slow preparation process, right? So, and which in turn leads to a pretty extended procedure time. And the critical one is the availability of the pathology resources. Some hospitals are really blessed that they have the pathologists, the cytopathology team coming down, and then they can make the specimen slides instantaneously, while in many cases, they do not have the luxury. For example, in one of the clinical sites that we go to, the physician is in the middle of a procedure, and then after the robot is all set up, they go in and then they call the pathologist like, hey, you know what, can you come down? And then the pathologist immediately is like, hey, I'm so sorry, I cannot come down today. You go ahead and then make the slides. So this is that kind of issue that happens not just in few sites, but in many sites at this point in time. So now the question is, how do we solve this problem? And that's exactly where the automated sample preparation comes in, right? So I'm sure you would have seen a cart like this in your clinical setup where they receive this and then they have these jugs of different stains that's there. And then they also have like a very sophisticated hair dryer where after you get the slides from the FNA, after it is smeared, they use a hair dryer and then they do these series of stains. So now imagine that all of these is getting replaced by a single system where your sample can be loaded and then you press the button and then the machine is going to completely prepare the slides. So I'm going to show you the video of our product, and then I'll talk through this one as the video is going on. As I mentioned, this machine does both the sample deposition as well as heating of the sample, as well as it has the staining done in one single go. So it has all of the stains incorporated. So the staining that is being done is the Romanowski staining, or commercially it's a Diffquick stains. So it has all of the required stains that's in there. And then it comes with these consumables that you're going to see over here. And what you do is, so these three consumables are there. So we have developed this machine such that it's easy for anyone with limited to no training on cytopathology to use this one. So each and every time you insert the consumable, you can immediately see the lights go on. So this actually shows that this is the direction to insert the consumables and the order of consumables. So as soon as you insert the slides, you then insert these what we call as a specimen hoods. The reason why we came up with this is, and this is critical during the COVID and in other cases where we saw that as people were doing rapid on-site evaluation, many of the pulmonologists were actually doing rapid on-site and were part of the FNA process, they ended up getting COVID. The reason is because you prepare the slides and then you are blowing air onto the slides to heat it, and all of these cells fly around and then eventually they get COVID as well. In fact, one of our board members, who is also a renowned physician, a renowned pulmonologist here in the United States, got COVID, and he vouches that he got it when he was in the procedure, right? So now what we have here, which I'm showing, is what is called as a pod, and your FNA sample that comes in, it goes into this pod, and then you go ahead and then insert this sample as a cassette inside. And once this is all done, you go ahead and then press the start button. And within less than 1.5 to 2 minutes, it's going to make you two slides that is similar to what is happening right now. One of the slide is going to be Diffquick stained or Romanowski stained, while the other slide is going to be fixed in ethanol so that the pathologist can actually take it there to their lab and then go ahead and then do the subsequent processes. So as I mentioned, this is our first product that focuses on the sample preparation. I'll talk about subsequent ones that's going to be there. But just from a high level, I just want to recap on what this particular instrument does, right? It's a very compact and a mobile system. that combines both the deposition and the staining into a single compact unit, right? So this can be placed in a pulmonology suite, bronchoscopy suite, or suites, etc. It's compact enough to be placed. And it can be placed, in, most importantly, on a rose cart. And I don't know if your clinical site uses something like a rose cart, you can pretty much use it in there. And the most important thing is you don't need this external dryer and everything. It has an integrated heating element for quick slide drying. And the most important advantage of this machine is it uses a fresh stain each and every time. So if there are some pathologists in this particular webinar, they can realize that one of the major challenges with the existing ROSE processes, when they are making the slide, let's say on a Monday morning, the stains are really fresh. It's really good quality of slides are seen, but then the stains deteriorate over time. So by the time Wednesday or Thursday comes in, the quality of the staining is really bad, which makes it really hard for them to visualize these slides. So what this machine does is it dispenses a fresh stain, a small amount of fresh stain each and every time. And this is something that we have gotten a lot of really amazing feedback on, where our stain quality is really sharp and it's much better than what is there right now. And as I've been saying, it performs diff quick staining, which is a Romanowski staining, and everything is done in less than 2 minutes. And the most important thing is it avoids any kind of a cross contamination between the samples. This is extremely critical. You don't want to confuse if the samples are coming from 4R or 4L, etc. You want to make sure that there is no cross contamination whatsoever happening, which is a huge challenge right now, where you will not be able to control for the cross contamination. So with this one, let me very briefly talk about a multi-center clinical study that we did. I'll talk about that particular the study data, and then I'll talk a bit more about some of the slide qualities that we have been getting. So we did collect samples on around 200 patients or so who are undergoing EBIS TBNA across 5 different institutions. These are renowned institutions, as you might already recognize from here. First Health, Mayo Clinic, University of Chicago, Loyola University, and of Fairfax. The way in which we did this study was we wanted to compare whether our preparation, which is the preparation using the automated machine, is as good as the conventional standard of care. And again, the conventional standard of care, the slides are being made by a cytotechnologist or cytopathologist with years and years of experience. So our question is, can an automated machine even make the slides that's as good as a renowned cytopathologist, right? So we split the samples into 2. And then what the cytopathologist did was they compared the samples obtained from both the standard of care as well as the automated preparation. And then they look for their characteristics that they specifically look for during the rapid on-site, right? So they look for the nuclear quality, the cytoplasmic quality, the presence of debris. And the most important thing is how easy is it to diagnose the slides that's actually coming from our preparation method. And the most important thing is the creation of the monolayer. So, and then for each and every of these metrics, they assign a score between 1 and 3, with 1 being the worst and 3 being the best. So with this one, so this is a study summary, right? So, and I don't want to go through each and every metric over here, but what you can see here is the instrument prepares the sample, which is comparable to that of the standard of care. And most importantly here, I'm re-emphasizing it, the standard of care is being made by a pathologist or the cytotechs who with years and years of experience from these world-renowned clinical sites, right? The most important metric where our preparation method leads to a much better quality is on monolayer creation. So ours, the slides that's coming out of our machine creates a much better monolayer compared to that of the standard of care. Why is this important, right? In the future, our goal is to integrate our automated machine into an AI-based diagnostic platform, as a result of which you want to have a machine that creates a nice thin monolayer, which enables getting to the diagnosis far more quick. So with this one, let me go to the overall study summary. These are just 2 examples of the slide or few images that's coming from the standard of care preparation and the ASP device, right? So as I said before, our study results suggest that the slides that are prepared using the automated system is comparable to that of those prepared using the standard of care method. And of course, it creates a better monolayer compared to that of the standard of care. As we go in and then as we talk to people, one of the issues that we came across is on sample homogenization. So right now, if I understand the way in which it is done in multiple clinical sites is, when you insert an FNA needle, you take the sample and then you put a couple of drops onto the slide where the rapid on-site evaluation is being done, while the remaining sample goes on to for molecular analysis. So what it creates is the sample inhomogeneity, or there is a huge variability in where exactly the diagnostic cells are present, right? So, and this actually leads to a lot of confusion because in many cases that we have seen here in the clinical sites in the US, they really get an amazing rapid on-site slide. However, when they send the sample over to molecular, they come back and then say, hey, you know what, unfortunately, there isn't enough samples that's actually present in there. And what is the reason for this? The reason is that the cells, the diagnostic cells, are not equally distributed across the entire length of the FNA needle. Sometimes the diagnostic cells are present in the front of the needle, sometimes it's in the back of the needle. And because of all of these things, what happens is you eventually have to go on to make additional passes for molecular analysis. And even that is not perfect, because when you keep on making additional passes, these additional passes might just end up getting blood. So what it essentially happens is there isn't an exact replica of what is there in the rapid on-site slide and what is there in the molecular. So in order to circumvent or in order to avoid that issue, what we have is a specialized vial that you're seeing over here, and that enables you to completely homogenize the sample. So the workflow, the way in which it works is, after you get the sample from the FNA needle, you put all of the sample from the FNA needle into this particular vial. And then you go ahead and then massage the vial. You put a couple of drops or so into the machine that I showed you earlier. And then the remaining sample goes in for the molecular analysis. So let me show you a video to have a better understanding of this. So for example, here, the sample from the FNA needle comes in, and all of the sample goes into the sample vial. And then let's say your respiratory tech or the nurse in there would gently massage it to equally disperse the sample. And then they load the sample into the pod, which can then be inserted into the machine. So this pod gets inserted into the machine, and then the remaining sample that's left in the vial directly goes in for the molecular analysis. If you know for the fact that the slide that is in the rapid on-site shows diagnostic cells, you can be rest assured that even the samples that's there in the vial that is sent for molecular analysis is going to contain cells as well. Okay, so with this one, so we have been getting a lot of critical feedback on the sample homogenization, because what we have seen is close to 20 to 30% of the samples that's being sent for molecular analysis comes back as inadequate. So, and this overall workflow using both our instrumentation as well as using the sample vial would dramatically decrease both this kind of the repeats that you have to do for the molecular, as well as as well as decrease your procedure time overall by somewhere between 15 to 30 minutes per bronchoscopy procedure. So with this one, let me show you some of the slides that are prepared using the automated machine. So you can really appreciate the monolayer that is coming from this particular machine and the stain quality that is seen in here, right? So this is just a few examples of samples that are prepared using the lung FNA. So it shows a really nice thin monolayer that's in there that shows critical diagnostic cells that's in here. Of course, you're seeing few cancerous cells over here. And then again, the stains are pretty sharp, as you might recognize. And the critical thing is, since it's a nice thin monolayer, your pathologist can very clearly see the nucleus and the nuclear details and nucleolus inside the cells, etc. So as I have been saying, this machine is a sample. It's a sample preparation platform, right? It can prepare samples from lung, pancreas, thyroid, etc. So these are just examples of the preparation that's obtained from the pancreatic ductal epithelium. So that again shows a nice thin monolayer of cells that's in there. And this is the one from renal cortex with showing nice diagnostic cells over here with red blood cells in the background. And this is an example of the thyroid follicles that's in there with a nice thin monolayer of cells. And these are the reactor lymph nodes, which I'm sure you might have seen several times on your own slides. So now the question is, where do we go from here, right? So as I said, this is an automated machine. And right now, this machine is FDA approved here in the United States. And it is currently in 10 different clinical sites here in the US. And we are slowly placing these units in different clinical sites here in the US. And recently, we launched it. We are currently getting the CE mark in Europe, and we hope to launch the product in Europe in the next couple of months or so. So in the US and in the Europe, by the end of the year, we hope to place these units in different clinical sites. So with this one, now the question is, where do we go from here, right? So our vision, as I mentioned earlier, is this is just a step one towards it, right? So where we have an automated machine, where we think this automated machine is going to standardize the entire workflow. But what we also want to do is we want to create an efficiency in the overall workflow of rapid onsite, right? So because in many places, the pathologist would not be able to come down. So now the question is, we want to combine the automated machine with the remote microscope. So we are working with few remote microscope players, both in the Europe as well as in the United States, in order for them to place both these units together in the bronchoscopy suite, so that as and when the machine comes up, as and when the slides come out of our machine, they can immediately be loaded into a remote microscope, so that your cytopathologists don't even have to come down. They can look at the slides remotely and then give you an adequacy feedback. Or in fact, they can also provide a diagnostic feedback instantaneously. So that way you can go on to speed up your overall procedure. So this is our step one towards it. And of course, as we have been talking about, there is certainly a huge momentum here in the US for the same day diagnostic treatment, right? So, and this is something that we are working on, where our machine provides the standardization that is required for future artificial intelligence-based diagnostic application, right? And again, I'm not promising anything at this point. It's going to be at least 2-3 years or so. What we envision is this automated system will be the first step towards completing the entire automation of the rapid process, complete from the adequacy and the diagnosis using the AI. So for example, let's say once the slide is inserted, the machine would prepare the slides, and then it would image the slides, and then it would tell you there are cancerous cells that's actually present in there, or it's a completely a normal benign cells, as a result of which you can go on to guide your next step of your overall workflow process. So currently there are further studies that are being performed to study this effect of automation in reducing the overall time. Our preliminary data shows that it is able to reduce it by at least 15 minutes or so of your overall procedure time. And there are more studies that are currently being done. So with this one, let me stop here to see if anyone has any questions.
Thank you, Dr. Subramanian, for this lucid presentation. And I'm joined with Dr. Sumita. She's an intervention pulmonologist practicing at Medipulse Hospital in Jodhpur. So we have some questions here on the message and also from our own unit. Like Sumita.
Yes. Hello, Dr. Subramaniam. Hello, Dr. Hari. And thank you all the speakers as well.
Yeah. So Dr. Subramanian, like what is the time to prepare each slide? Like how much time it will take, and how many slides for each test, like each cartridge, how many slides it can make? Can you just elaborate on that?
Yeah, so it takes anywhere between 1.5 minutes to 2 minutes to prepare a slide at this point in time. And the reason why there is this variability is it depends on the mode of preparation that you're using. So for example, if you want to just stain the slide, that's one thing, versus if you were to both deposit as well as stain the slide. We do get a lot of questions, as let's say primarily from the pathologist. Hey, I can actually make these slides within 30 seconds, right? So which is true. There is a vast majority of the people who think that they can make the slides within 30 seconds, but in reality, that's not the case, right? So we have timed how much time they actually take to make a slide. It comes anywhere between a minute and a half or so, right? And again, there could be some pathologists out there who are extremely quick, but at least according to the protocol that these stain companies or these dye companies give out, the overall protocol time itself is 1.5 minutes, right? But again, that's where the variability that we are talking about. So that's to answer your first part of the question. Yes, the machine is, it does this in a very systematic manner, right? So, and it's going to be the same time regardless of who or does it, right? Because some of your pathologists might be really quick, while some pathologists will be really slow, while this one, it's pretty consistent all across. The second question is the cartridge. So except those three consumables that we talk about, all of the dyes and everything, it is all your own dyes, right? So your clinical site, whoever engages with, you can use your own dye platform. But we have developed a machine where the container can hold enough dyes to make samples for around 120 or so passes, so which is typically 4 or 5 patients in a given day. So our recommendation is at the end of the day, after you have many passes, you go ahead and then check it. And then if you need more dyes, you go ahead and then manually fill it up.
Yeah, well, sometimes we do, our pathologists are very generous. They come for all our ROSE procedures, but sometimes we also try to do ROSE by ourselves, and we know the difficulties what happens when we make slides. Sometimes when I give my bronze sister to do the slides, sometimes they wash away all the dye that is there on, and then they put the sample and we see no material. I think that problem is definitely answered with this. Second is like how much sample quantity is needed from each FNA. Like sometimes the aspirate depends on the type of node. Sometimes we don't get any like adequate quantity to make a slide. So how much of the sample is really needed to make a proper quality slide in this platform?
Excellent question. So this one can utilize as small of a sample as a small drop of sample, right? So because we understand that there are going to be a huge variability in the amount of sample that you get. Sometimes you get just like a very small drop. Sometimes you get a lot of sample in there. So that's where this vial, which I was actually showing you, it helps you, right? So if you just get one drop of sample, you put it in there, you massage it and then load it. And then if you have a lot of sample, you still do the same thing. But the way in which the machine has been developed is it constantly takes the same volume of sample, because our goal is to only use a very small sample for rapid on-site, because we wanted to preserve as much sample as possible for downstream pathology analysis and molecular analysis, etc. So that's where we want to do. We certainly do not want to have all of the sample goes into making a row slide, which means that you do not have enough sample whatsoever for molecular. We understand that more and more of these diagnosis is going into the molecular route. So that's where we are making a very, very small sample, takes a very small sample for the automated analysis.
So one more question from the audience is like, what would be the approximate cost per each test when we use this platform currently in the United States?
Yeah, in the US, it is, yeah, I mean, my investors would be, would not be very happy if I were to divulge the pricing information over here. But let me tell you, we currently have a very friendly family and friends pricing options for some of our initial sites and everything, which is going to be lower. And our goal is to make the pricing would not be the limitation for you to implement this instrument in your clinical setting, right? So, but again, we'll be more than happy to discuss about the pricing. But we have made sure that the pricing is lower than the current reimbursement process of rapid onsite in the United States.
And right now we sometimes get like these cryobiopsy samples, and then we make some imprints of these cryobiopsies. Does this system support that? Like if you get some imprint from the cryoprobe?
Excellent question. Yes. So this instrument can prepare samples from cryobiopsies, touch biopsies, brushings, and of course the FNA. So in the case of the FNA, it uses both the deposition as well as staining, where you just take a drop of liquid and then put it into the machine. While in the case of a cryobiopsy, where you do a touch imprint or a touch prep biopsy or a touch biopsy or a brushing, since the slides are already having the cells in them, you use a different mode, which is the stain only mode, which you can utilize it and then you can run it. So yes, the machine can be operated in all types of samples that you get during a bronchoscopy procedure.
Okay, Sumita, do you have any questions from your end?
Yeah, so in follow up to the question that Dr. Hari asked earlier, that how much amount of sample you will put in that solution to get the slide, I have this doubt, like you said, very small amount of sample is needed. So we put like very small from that pipette. What if those slides are negative and you actually have some positive slides in the rest of your container that you said? In that case, you're actually missing on the rows. So is there like any other way to understand?
Yeah, that's a really great question. And this is something that we have been doing some studies over here. And this kind of variability happens right now, right? Because for example, in your FNA needle, you take the sample, you put a couple of drops onto the slide, and then you still have a lot of sample that's left in the needle, and then you take it to the molecular. That variability is certainly there. It exists right now in the current standard of care process. What we are trying to do is, even though you might only take one drop of sample from this, let's say, for example, you fill up, as an example, you fill it up with 60 microliters of sample here, right? And then you massage this, and then you only take 10 microliters of the sample from the vial and then put it into the machine. Yes, there would be a very, very small percentage probability that the sample or the diagnostic material might not be there in that 10 microliters. It might be in the remaining 50 microliters. But that probability is very, very small. The reason why it is small is you are completely homogenizing the sample, right? You are completely massaging the sample, you are homogenizing it, where the cells are equally dispersed across the entire length of that 60 microliters. So when you're loading the 10 microliters of sample, as an example, when I told you, that would be a certain exact replica of the remaining 50 microliters that's going to be in there. And of course, there is going to be a difference if you only have, let's say, overall, you only have 10 cells to begin with, right? So if you only have 10 cells, diagnostic cells in there, yes, you would have that issue where the 10 microliters that you're loading into the machine is not going to be a replica of 50 microliters. But I'm pretty sure that your pathologist might not be happy with having only 10 cells in your overall FNA.
Right, but many times the sample is mixed with blood clots as well. So is there any way that that can also be an issue when we're using this?
Bloody
sample.
Hmm.
Yeah, it's a bloody sample and the blood clot. So all of those we have actually. So this particular vial, once you utilize it, you can actually see this one. It allows you to massage and then disperse the cells out of the blood clot, because that's something that you cannot currently do, because the blood clot sometimes contains a lot of diagnostic material in there. So this would actually enable you to disperse those cells off so that you can get it into the rapid onsite.
Right, great. Yeah.
So
and tested a lot of slides that are really bloody that's in there.
Dr. Subramaniam, like in the study you showed, like what are the real world challenges you faced? Like what is the means? Is there a difference in the diagnostic yield between the conventional Rose method, and though we are not looking at a diagnosis in Rose, what are the real world challenges that you had? The plus of this technology and what are the minuses?
Yeah, that's a great question. So As far as the diagnostic yield is concerned, there was a 95% correlation between the diagnostic yield that's obtained from this method as well as the standard of care method, right? So now with respect to the real world challenges that we are facing, a couple of them are, it's more of a workflow challenge, right? Because now who exactly is going to be operating this machine? Because if in many sites the cytopathologist doesn't come down, they see this particular machine in there, and then it's like, oh, you know what? I do not have to come down, so I'll have the respiratory text run this one, right? So we have had that happen in quite a few places, right? So which is, in the long term, it is a good news, but in the short term, it's like, hey, you know what? Who exactly is going to help? So that's where in many of the sites that we work with, we collaborate with both the pathologist as well as the pulmonologist together so that they know exactly who's going to be handling the machines, etc. The second one, which the real world challenge, again, that comes in between the pulmonologist and the pathologist is who exactly takes control of the machine, right? So for example, who's responsible for filling up to make sure that the dyes are there, etc. Again, this is where these are the initial challenges that we face because, for example, the pulmonary technicians or pulmonology nurses are not used to looking at these kinds of stains, and then it's like, hey, you know what, is it enough that's there? But that we feel is the initial challenge as we start the adoption process. And once it is there, and once we have a clear division of labor as to, hey, you know what, this is who is going to be responsible for this, we hope those challenges would be slowly mitigated.
I think that's a great technology. Why, in my personal opinion, we have a very good pathology collaboration, but I think the workflow definitely more than the intervention pulmonology, the pathologist would be more happy with this technology because of the time. Because sometimes what happens is we have 3 to 4 cases of EBUS a day, and then they have some frozen samples that has to be read immediately. So in that manner, I think they will be more than happy if we train, because this I think is a very simple technique where we can also just make the slides, prepare the slides by ourselves by the time our pathologists come and then read them, or if you are able to read our own things. And I think it's a great platform. Thank you so much, Dr. Subramanian. And I have one last question here. Dr. Sivatsa from Bangalore, he's asking, sometimes we flush our samples and the pathologist centrifuges these samples. So take the cellular content and uses that to make cell blocks. So will that be useful in this situation? Like, I mean, those samples where we can scrape from the slides and then send them for genetic analysis? Or once you make a slide, that's the end of the slide and you cannot extract any material from this. So what is your take on this?
Yeah, that's a great question. So the slide that is getting prepared here is exactly same as the slides that is getting prepared using the manual methods, right? So for example, yes, you can go ahead and then scrape the slides and then send it out for genetic analysis. You can use the sample that's coming from this particular vial to go ahead and then make the cell block out of it. You can directly send it out for molecular analysis for NGS, etc. So yes, you can do any and all analysis that you're currently doing with the manual method. You can do this with our automated machine as well. So now the question is, with respect to the scraping of the slides, etc., we work in close collaboration with a lot of cytopathologists here in the US. Our hope is, because they struggle a lot with that kind of a scraping method and then getting to the genetic analysis, etc. Our hope is this kind of a vial method that I was talking about would pretty much revolutionize the way in which they have to prepare the samples for the genetic analysis. So this would pretty much streamline it, because the reason why they are doing that scraping method is because they do not have the exact replica for the molecular. So while this one, we are hoping it would be a perfect replica of rows and the molecular.
Okay. Yeah, I think that answers most of the questions. Like, there was one question that was asking what is the sensitivity and specificity of this device compared with the conventional done rows.
Yeah, so this machine is not a diagnostic machine. So it is just an automated sample preparation machine. So there is no sensitivity and specificity per se here. So for example, it all depends on the sample that you put in there. If the sample is really bloody, the machine would make a really bloody slide. If the sample is diagnostic material is there, the machine would make a diagnostic or a slide with a diagnostic material. So the sensitivity and specificity will be same as that of your existing platform that you have.
Great information. So I think with that we will come to Sumita. Do you have any other questions?
I think no, most of the
I
questions
have
have been answered.
one last question, Dr. Subramanian, if you're okay with that. See, you're showing that vial, right? So if I
Mhm.
make two slides in my first pass, so I keep this vial inside, and then for the second pass, I do from a different station. Do I have to use a separate vial or the same vial? And does a new vial will incur new cost to the procedure, or how is it?
Yeah, it is true that you have to use fresh vials for each and every pass. And there is a reason for that, right? Because let's say, for example, if your first pass is just blood, and then you do not have anything, while your second pass, you're exactly in the lesion, and then you get the most enriched amount of sample. You do not want to mix up your first pass with the second pass, right? Because then you are just diluting the overall diagnostic cells. And that is the reason why our recommendation would be to use one vial per pass. And again, these vials are really cheap. So as a result of which, we do not want the cost of the vial to be uh, uh, I mean, a limitation for you to implement the procedure or the workflow.
And is there like you put more material in and that will smudge the smear, or it will take only a little amount from that as a fixed amount for every slide that you make from this container?
That's a major advantage of it. So even if you have 100 microliters of sample that's coming in, or if you only have like 10 microliters of sample coming in, the machine is going to make the slide very, very consistently. It's only going to take the constant volume of liquid every time, as a result of which it's not going to smudge. And that is one of the reasons why our machine makes a much higher content of monolayered slides compared to that of the standard of care. Because in the standard of care, we do see many of the slides where it gets smudged up and it is very hard, etc. That would not happen using our machine.
So I think you have answered most of the questions from the audience and also from the panel watching joining here. Thank you so much, Dr. Subramanian, for this
Thank you.
information what you gave us, and we are very eagerly waiting because it definitely helps my intervention pulmonary unit, whatever whether you do ROSE, all the studies may say that the diagnostic yield may not vary, but I think the processing time definitely makes a difference, not only for the IP but also for the pathology standpoint in my hospital. So what is the timeline like? When do you have plans to get this technology in India? And
Yeah, so in India, we are certainly looking for right partners to get there. So our current focus is certainly in US and in Europe. However, if you have any good partners that you suggest, I mean, when I say you, anyone in the audience who can suggest us, we would love to talk to them. And so that's what would be the limitation for us. We would love to launch this in multiple countries, and we have already seen a lot of interest in India and in other countries as well. So it's a matter of our prioritization and the other ones, because the regulatory hurdle, we think it's going to be very, very low for a product like this.
yes, and definitely we can provide a lot of information on the benign pathologies, especially tuberculosis, and a lot of granulomas from this machine you can expect when you come to India. So with that, I would like to thank Dr. Subramanian for his valuable time, and we come to the end of the question and answer session here, and then I would like to inform all the audience that the next webinar, the August episode, is going to be on the new navigation software that is coming up from Fujifilm in the bronchoscopy aspect. That will be Synapse 3D platform. So we have colleagues from Japan and myself. I will be discussing a few cases on Synapse navigation platform in our August episode from Yashoda Hospitals and the Academy of Bronchoscopy. Thank you so much for watching this, and I hope all of you got some new information on the automated sample processing for rows. Thank you, Dr. Subramanian and Dr. Sumita, for joining us, and all the audience.
Yeah,
Thank
thank you
you.
so much, everyone, for this. And thank you so much, Dr. Harikishan, for inviting me for this one. Thank you.
Thank you.
Thank you so much, Dr. Raghavan.